Going about your daily life, you must often respond rapidly and appropriately to your environment in order to function as a capable human. It is your nervous system that makes this possible. Walking down a city street, your eyes, ears, nose, and other receptors throughout the body receive a flood of sensory information, which is sent to your brain by electrochemical impulses racing down the sensory nerves that branch throughout your body. In the brain, that sensory information is sorted through almost instantaneously. Some is ignored, some placed in memory, while other information is responded to immediately. In responding, the brain sends electrochemical impulses via motor nerves to the effectors, the muscles of the hands, legs, feet, lips, vocal cords, etc., necessary to achieve the desired response. But how do nerve impulses travel through sensory and motor nerves to the brain? And how does the brain decide which stimuli to react to and which to ignore? Why are we conscious of some nervous system activities and not others? Before going on to answer these and other questions about the nervous system and looking in more detail at the structure and function of the brain and other parts of the nervous system, we must first understand something about the specialized cells that make up the entire nervous system, including the sensory and motor nerves, the spinal column, and the brain itself. The specialized cells that make up the nervous system are called neurons. A typical neuron consists of 4 regions: dendrites, cell body, axon, and synaptic terminals. Dendrites are the tangle of fibers that branch from the cell body. They convert information received from the outside world or other neurons into electrical signals. Electrical signals generated by the dendrites converge upon the cell body, which then adds up the various signals and decides whether to produce an action potential, the electrical output of neurons. In most neurons, a long, thin fiber called an axon extends outward from the cell body. The axons of different neurons are usually bundled together into nerves that emerge from the brain and spinal cord and extend throughout the body. Axons and the nerves they form can be very long. For example, there are axons that stretch from your spinal cord to your toes, making them the largest cells in your body. Axons carry action potentials from the cell body to synaptic terminals, which appear as swellings at the branched endings of an axon. At synaptic terminals, nerve signals are transmitted chemically. Most synaptic terminals contain a specific chemical or neurotransmitter that they release in response to an action potential. The synaptic terminals of a neuron may communicate with a gland, a muscle, the dendrites or cell body of another neuron. Unstimulated, neurons maintain a constant electrical difference or potential across their cell membranes. This potential, called resting potential, is always negative inside the cell and ranges from -40 to -90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative depending on the stimulus. If potential is made sufficiently less negative, it reaches a level called threshold, and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell restores its negative resting potential. The cell membrane of a neuron encloses cytoplasm with various ions dissolved in it. The neuron itself is immersed in a salt solution, the extracellular fluid. The ions of the cytoplasm consist mainly of positively charged potassium ions and large negatively charged organic molecules, such as proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up cell membranes, they must travel through channel proteins that extend through the membrane. In an unstimulated neuron, only potassium ions can cross the membrane, traveling through specific proteins called potassium channels. Although sodium channels are also present, in unstimulated neurons, they remain closed. Since only potassium ions can cross the membrane and potassium ions are more concentrated inside the cell, they diffuse out of the cell, leaving the large negatively charged organic ions behind. As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside. At some point, the diffusion of potassium ions out of the neuron due to concentration differences is balanced by the electrical attraction tending to pull them back inside. This is the point at which neurons reach resting potential. Reaching resting potential in this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of the potassium ions initially inside a neuron must leave to create a resting potential of -60 millivolts. But it is action potentials, not resting potentials, that carry information through a nervous system. Action potentials, starting at the point where the axon leaves the cell body, travel like a wave of positive charge, undiminished in magnitude, along the axon to the synaptic terminal. Immediately after the action potential passes any point along the axon, the negative resting potential is restored within the axon. At rest, a neuron is something like a loaded gun, ready to fire if the trigger is pulled. In a neuron, the explosive charge is the high concentration of sodium ions outside the neuron. The trigger is a set of membrane proteins called sodium channels. Sodium channels are closed in resting neurons, but open rapidly once a condition called threshold is reached. The opening of sodium channels allows positively charged sodium ions to flood into a neuron, make its interior momentarily positive. Threshold is triggered in a neuron by chemical signals received from neighboring neurons. A short time after threshold is reached, the sodium channels close again, while potassium channels open. Potassium ions are now driven out of the cell by both diffusion and electrical repulsion to the positively charged sodium ions. Almost immediately, enough potassium ions are driven out of the neuron to cause resting potential to be reestablished. Action potentials are all or none events. Thus, no action potential is initiated unless threshold is reached. But if threshold is reached, a full magnitude potential travels the entire length of the axon to the synaptic terminal. Only a tiny fraction of the potassium and sodium ions in and around each neuron is exchanged during each action potential. However, after a few thousand action potentials, the sodium and potassium concentration gradients across the neuron membrane would be lost, if not restored by active transport molecules in the cell membrane called sodium-potassium pumps. Sodium-potassium pumps use energy from ATP molecules to pump sodium out and potassium in and maintain the concentration gradients necessary to power the diffusion of these ions across the cell membrane of a neuron. In order for a neuron to conduct an action potential along its axon to its synaptic terminal, the action potential must not diminish in magnitude and die out along the way. Neurons maintain the magnitude of action potentials by renewing them at successive points along the axon. While part of the positive charge of an action potential leaks out the cell membrane as it travels down the axon, enough remains to cause regions further down the axon to reach threshold, open their sodium channels, and renew the action potential. This process continues along the entire length of the axon. Meanwhile, once the action potential has passed a given section of axon, its sodium channels close, its potassium channels open, and the resting potential is reestablished. It is often important that action potentials travel rapidly, for example, to avoid a sudden danger or threat. However, the opening and closing of ion channels during action potentials is relatively slow. Therefore, for a signal to travel as rapidly as possible, as few channels as possible should be opened and closed. In vertebrates, axons that need to conduct rapidly, such as those that carry signals to muscles used in pursuit or escape, are wrapped with insulating layers of myelin, which are interrupted at intervals by exposed areas called nodes of Ranvier. Myelinated axons transmit signals extremely rapidly because ion channels are concentrated only at the nodes. When an action potential occurs in a myelinated axon, the positive charge that enters the axon can't leak out because of the myelin, but instead travels almost instantaneously to the next node, where channels open and a new action potential is initiated. This process continues as the action potential travels down the axon. The transmission of an action potential along a myelinated axon is called saltatory conduction, literally jumping conduction, since the action potential appears to jump from node to node as it speeds down the axon. When an action potential reaches the synaptic terminal of the axon, it encounters a region called a synapse where two neurons come close together but do not touch one another. A minuscule gap, called a synaptic cleft, separates the synaptic terminal of the first or presynaptic neuron from the dendrite or cell body of the second or postsynaptic neuron. The arrival of an action potential at the synaptic terminal causes the synaptic terminal to become positively charged and release neurotransmitters into the synaptic cleft. The neurotransmitters diffuse rapidly across the synaptic cleft and bind to receptors in the membrane of the dendrites or cell body of the postsynaptic cell. Each type of receptor in the postsynaptic membrane binds to a specific type of neurotransmitter, and after binding to a neurotransmitter, the receptor causes specific types of ion channels in the postsynaptic membrane to open. As ion channels are opened, ions flow across the cell membrane of the postsynaptic neuron along their concentration gradients. This flow of ions into the postsynaptic neuron causes a postsynaptic potential in the dendrites or cell body. Depending on the channels open and the ions that flow in, synaptic potentials can be either excitatory, making a neuron less negative inside and more likely to fire an action potential, or inhibitory, making it more negative and less likely to fire. An excitatory postsynaptic potential is called an EPSP. An inhibitory postsynaptic potential is called an IPSP. A synapse that produces EPSPs in the postsynaptic cell is called an excitatory synapse, while a synapse producing IPSPs is an inhibitory synapse. Postsynaptic potentials cannot travel far in a neuron. After a few millimeters at most, the ions leak back across the membrane and the signal is lost. However, postsynaptic potentials travel far enough to reach the cell body, where they determine whether or not an action potential will be produced. The dendrites and cell body of a single neuron can receive EPSPs and IPSPs from the synaptic terminals of thousands of presynaptic neurons. The postsynaptic potentials produced by different presynaptic neurons are added up or integrated in the cell body of the postsynaptic neuron. The postsynaptic cell produces an action potential only if the excitatory and inhibitory potentials, when added together, raise the electrical potential inside the neuron above threshold. Individual neurons and their language of action potentials, when tied into networks of neurons called nervous systems, allow even simple animals to perform an impressive array of complex behaviors. In order to perform its duties, a nervous system must be able to perform 4 operations: determine the type of a stimulus, signal the intensity of a stimulus, integrate information from many sources of stimuli, and initiate and direct response to the stimuli. To identify the type of stimulus, light, touch, sound, etc., the nervous system monitors which neurons are firing action potentials. The brain identifies action potentials occurring in the axons of optic nerves as information about light, action potentials in the olfactory nerves as odors, and so on. Since all action potentials have the same magnitude and duration, no information about the intensity of a stimulus, like the loudness of a sound, can be encoded in a single action potential. Instead, intensity is coded by the frequency at which individual neurons fire action potentials and the total number of neurons firing action potentials. The more intense the stimulus, the faster neurons fire and the greater number of neurons firing. The brain is continuously bombarded by a variety of internal and external sensory stimuli which it must filter, determining which stimuli are important and what action to take in response. Nervous systems integrate information through convergence, as many neurons funnel their signals to fewer neurons. For example, many sensory neurons may converge into a smaller number of brain cells. The brain cells add up the postsynaptic potentials resulting from the synaptic activity of sensory neurons, and depending on their relative strengths and other internal factors such as hormones or metabolic activity, produce appropriate outputs. The output of integrating cells initiates response, which may involve many parts of the body and requires divergence, the flow of electrical signals from a relatively small number of decision-making cells onto many different neurons controlling muscle or glandular activity. Most behaviors are controlled by neuromuscular pathways composed of 4 elements: sensory neurons that respond to a stimulus, association neurons that decide what to do, motor neurons that receive instructions from the association neurons and activate the muscles, and effectors, usually muscles or glands, that perform the behavior. Reflexes are the simplest behaviors. The withdrawal reflex, which moves a body part away from a painful stimulus, is a good example, using only 1 neuron of each type. Reflexes of this sort do not require intervention by the brain, although associated pathways do inform the brain of the stimuli and perhaps trigger more complex behaviors. Nearly all animals are capable of more subtle and varied behavior than can be accounted for by simple reflexes. In principle, more complex behaviors are organized by interconnected nerve pathways that provide several types of sensory input, along with memories, hormones, etc., that converge on a set of association neurons. By integrating the postsynaptic potentials from several sources, the association neurons decide what to do and activate the appropriate muscles and glands via the firing of motor neurons. The human nervous system has 2 major parts: the peripheral nervous system and the central nervous system. The peripheral nervous system consists of sensory and motor nerves that link the brain and spinal cord to the rest of the body, including muscles, sensory organs, and the organs of the digestive, respiratory, excretory, and circulatory systems. Sensory neurons carry sensory information to the central nervous system from all over the body, while motor neurons carry signals from the central nervous system to organs, muscles, and glands. The motor portion of the peripheral nervous system consists of 2 parts: the the somatic nervous system, and the autonomic nervous system. The somatic nervous system's motor neurons synapse on skeletal muscles and control voluntary movement. Their cell bodies are located in the gray matter of the spinal cord, and their axons go directly to the muscles they control. The autonomic nervous system's motor neurons synapse on the heart, smooth muscle, and glands, and control involuntary responses. The autonomic nervous system is further divided into the sympathetic and the parasympathetic nervous systems, both of which generally make synaptic contacts with the same organs, but produce opposite results. The sympathetic nervous system acts on organs in ways that prepare the body for stressful or high-energy activity, such as performing before a large audience or readying for a fight. During such fight or flight activities, the sympathetic nervous system curtails activity of the digestive tract, redirecting much of its blood supply to muscles in the arms and legs. Heart rate accelerates, the pupils dilate to admit more light, and the lungs' air passages expand to accommodate more air. Stimulation of the parasympathetic nervous system has the opposite effect. The digestive tract becomes active. The lungs, air passages, and the pupils of the eyes constrict. The heart rate slows, and the body in general becomes more relaxed. The central nervous system, which consists of between 50 and 100 billion, primarily association neurons in the brain and spinal cord, is the integrating portion of the nervous system, where sensory information is received and processed, thoughts generated, and responses directed. The brain and spinal cord are protected by the skull and vertebral column. Beneath the skull and vertebrae lies a triple layer of connective tissue called meninges, which is filled with cerebral spinal fluid and serves to cushion the brain and spinal cord from physical impacts. The spinal cord is a neural cable about as thick as your little finger that extends from the base of the brain to the hips through the vertebral column. Between the vertebrae, nerves called dorsal roots and ventral roots arise from the dorsal and ventral portions of the spinal cord respectively. In the center of the spinal cord are neuron cell bodies, which form a butterfly-shaped area of gray matter. These are surrounded by bundles of axon, called white matter, owing to their white insulating myelin coating. The spinal cord relays signals between the brain and the rest of the body and contains the neural circuitry for certain behaviors, including, as we saw earlier, reflexes. The function of some of the parts of the spinal cord are illustrated by a simple spinal reflex, the pain withdrawal reflex, which involves neurons of both the central nervous system and the peripheral nervous system. The cell bodies of sensory, in this case pain, neurons from the skin are found just outside the spinal cord in the dorsal root ganglia. Both association and motor neuron cell bodies are found in the gray matter in the center of the spinal cord. Association neurons for the pain reflex not only synapse on motor neurons, but have axons extending to the brain. Signals carried along these axons alert the brain to the painful event. The brain, in turn, sends signals down axons in the white matter to cells in the gray matter that can modify spinal reflexes. For example, with sufficient motivation, an individual can suppress the pain withdrawal reflex and expose themselves to the flames of a burning building in order to rescue a child. In addition to simple reflexes, fairly complex activities are also controlled by the spinal cord. For example, all the neurons and interconnections needed to walk and run are found within the spinal cord. The role of the brain is simply to initiate and guide the activity. The advantage of this arrangement is increased speed and coordination, since neural signals do not have to travel to the brain and back to move the legs. But our most complex behaviors are initiated and controlled by the brain. Embryologically, the vertebrate brain begins as a simple tube, which soon develops into three parts: the hindbrain, midbrain, and forebrain. It is believed that in the earliest vertebrates, these three anatomical divisions were also functional divisions. The hindbrain governed automatic behaviors, such as breathing and heart rate. The midbrain controlled vision, and the forebrain largely dealt with a sense of smell. However, in mammals, and particularly in humans, the brain regions are significantly modified. Some have been reduced in size, and others, especially the forebrain, greatly enlarged. In humans, the hindbrain consists of the medulla, the pons, and the cerebellum. The medulla is very much like an enlarged extension of the spinal cord. Like the spinal cord, the medulla has neuron cell bodies at its center, surrounded by a layer of myelin-covered axons. The medulla controls several automatic functions, such as breathing, heart rate, blood pressure, swallowing, vomiting, and digestion. Certain neurons in the pons, located above the medulla, are also involved in regulating automatic functions, such as the rate and pattern of breathing, while others appear to influence transitions between sleep and wakefulness and between the various stages of sleep. The cerebellum is crucially important in coordinating movements of the body. It receives information from command centers in the higher conscious areas of the brain that control movement and from position sensors in muscles and joints, as well as information from the visual systems and motion sensors in the inner ear. By comparing command center orders with information from the position and motion sensors and the visual system, the cerebellum produces smooth, accurate body motion. The midbrain is extremely reduced in humans. However, an important relay center, the reticular formation, passes through it. The neurons of the reticular formation extend all the way from the central core of the medulla through the pons, the midbrain, and on into lower regions of the forebrain. It receives input from virtually every sense and every part of the body, and from many areas of the brain as well. The reticular activating formation plays a role in sleep and arousal, emotion, muscle tone, and certain movements and reflexes. It filters sensory inputs before they reach conscious regions of the brain, although the selectivity of the filtering seems to be set by higher brain centers. Through a combination of genetically determined wiring and learning, the reticular formation decides which stimuli require attention. Important stimuli are forwarded to the conscious centers for processing, while unimportant stimuli are suppressed. The fact that a mother wakens upon hearing the faint cry of her infant, but sleeps through loud traffic noises outside her window, testifies to the effectiveness of the reticular formation in screening inputs to the brain. The forebrain, which can be roughly divided into three functional parts, the thalamus, the limbic system, and the cerebrum, receives sensory information, processes it, and initiates actions. It is in the forebrain that the most sophisticated neural processing occurs. Intricate integrating networks and sensory and motor pathways in the forebrain allow pattern and image formation, as well as functions such as memory, learning, and emotions. Most of the sensory and neural information directed to the cerebrum from the auditory and visual pathways, the spinal cord, the brainstem, and the cerebellum is first channeled through the thalamus, which sorts through the information and sends it to the appropriate area of the cerebrum for interpretation and response. The thalamus also receives and sorts information from what is often described as the emotional and memory center of the brain, the limbic system. Anatomically, the limbic system is a diverse group of structures that form an arc between the thalamus and the cerebrum and work together to produce our most basic and primitive emotions, drives, and behaviors, including fear, rage, tranquility, hunger, thirst, pleasure, and sexual response. The assigning of emotional value to an event is in turn used by other parts of the limbic system to form memories and assist in learning. The limbic system includes the hypothalamus, portions of the thalamus, the amygdala, and the hippocampus. The hypothalamus contains many different clusters of neurons, including neurosecretory cells that release hormones. Through its hormone production and neuroconnections, the hypothalamus controls body temperature, hunger, the menstrual cycle, water balance, and the autonomic nervous system. In addition, stimulation of specific areas of the hypothalamus elicits emotions such as rage, fear, pleasure, and sexual arousal. Emotional values are assigned to our experiences by the amygdala, which is also involved in orchestrating appropriate behavioral responses to environmental stimuli. The amygdala receives input from many sources, including the areas of the cerebrum's cerebral cortex that handles our senses of smell, touch, hearing, and vision. Some neuron clusters in the amygdala produce sensations of pleasure, punishment, or sexual arousal when stimulated, while the stimulation of other areas reduce or enhance aggressive behavior. Humans who have been conscious during the electrical stimulation of their amygdalas report having feelings of rage or fear as a result of the stimulation. Next to the amygdala lies the hippocampus. Like the amygdala and hypothalamus, stimulation of different portions of the hippocampus can elicit behaviors that reflect a variety of emotions, including rage and sexual arousal. There is also substantial evidence that hippocampus plays an important role in learning and the formation of long-term memory. Some of the evidence includes the fact that the hippocampus is a site of extreme electrical activity during the learning process, and that individuals with extensive hippocampal damage are unable to form long-term memories. Though the process of long-term memory formation is poorly understood, the hippocampus apparently takes short-term memories formed by transient electrical activity and converts them to long-term memories by creating structural changes in the neurons and synaptic connections assigned to retaining a given memory. It appears memories processed by the hippocampus are stored at numerous locations in the cerebral cortex, though others have suggested that memories, like holograms, are both everywhere and nowhere at the same time. Both the amygdala and the hippocampus are, as we saw earlier, tied into various sensory areas in the cerebral cortex, but they are also tied to an area of the cerebral cortex associated with learning, reasoning, and personality, the prefrontal cortex. The prefrontal cortex apparently consults the amygdala, hippocampus, and other parts of the limbic system to obtain information about the emotional content of a situation or memory before making a judgment about a course of action. The notorious procedure known as the frontal lobotomy, used in the 1940s and 50s to calm uncontrollably emotional and violent patients, severs the connection between the prefrontal cortex and the limbic system, leaving a docile, lethargic patient literally cut off from their emotions. Fortunately, frontal lobotomies are seldom, if ever, performed today. In humans, by far the largest part of the brain is the cerebrum, which makes up about 80% of the mass of the adult human brain. Anatomically, the cerebrum is split into two halves called cerebral hemispheres, which communicate with each other via a large band of axons, the corpus callosum. The cerebral hemispheres are sophisticated information processing centers. In general, each hemisphere controls the motor and sensory functions of the opposite side of the body. Thus, the left cerebral hemisphere controls the motor function of and receives sensory information from the right side of the body, while the right hemisphere controls and communicates with the left side of the body. Also, one of the two hemispheres usually predominates in the carrying out of a given mental process. For example, The left hemisphere normally dominates in terms of speech, language, and mathematics, while the right hemisphere generally predominates in creative processes, spatial perception, recognition of faces, and the ability to recognize and express emotion. The corpus callosum of females is larger than that of males, suggesting that the two hemispheres communicate more in females than males. This agrees with studies that indicate that the hemispheres of male brains are more specialized than those of females, though it appears that in both sexes, either hemisphere can take over some of the functions of the other if forced to by circumstances such as injury or disease. Tens of billions of neurons pack into a thin layer on the outer surface of the cerebral hemispheres to form the cerebral cortex we looked at earlier. The cerebral cortex is often referred to as gray matter, as its high concentration of neural cell bodies give it a gray appearance. Its distinctive folds, called convolutions, evolved to increase the cerebral cortex's surface area. Functionally, the cerebral cortex is divided into 4 lobes: the frontal lobe, the temporal lobe, parietal lobe, and the occipital lobe. Each of these lobes has numerous areas that carry out specialized functions. For example, the motor cortex, which lies along the frontal lobe's boundary with the parietal lobe, is largely responsible for sending commands to skeletal muscles in response to sensory information. The sensory information to which the motor cortex responds is generated by touch, pain, pressure, and temperature receptors throughout the body that first relay their messages to the somatosensory cortex, which lies directly across from the motor cortex in the parietal lobe. The proportion of motor or somatosensory cortex devoted to a particular part of the body is correlated with the importance of that body's part in perceiving and reacting to the external environment. For example, our hands are much more important in perceiving and reacting to our environment than is our torso, and, as a result, the proportion of both the motor and somatosensory cortex devoted to the hands is much larger than that devoted to the trunk of the body, even though the torso of the body has much more surface area. The division of mental labor that exists between the 4 lobes of the human brain is further illustrated by looking at images of brain activity produced by PET scanners, as health subjects carry out a number of language-based activities. As individuals try to generate words with which to express themselves, brain activity is concentrated in the frontal lobe of the cerebral cortex, while as they try to listen to others, brain activity is focused in the hearing and auditory association areas of the temporal lobe. Individuals looking at words generate the most brain activity in the visual association area of the occipital lobe, while the majority of brain activity in individuals reading words occurs in the parietal lobe. Though devices like the PET scanner offer amazing insight to the activities of the human brain, science is still far from a complete understanding of how billions of neurons in the human brain interact to produce great pieces of music or visual art, or how the human brain developed the language of mathematics to accurately describe the complex events that occur in the physical world through simple symbols and numbers. The depth and vastness of the human mind, its ability to remember and to dream, have led some people to refer to it as a 3-pound universe. Though we have come to understand much about how neurons fire and how reflexive responses are generated by stimuli, one is left to believe, or at least hope, that, like the universe from which it was derived, the human mind and how it works will always be shrouded in a little mystery.